So I would next like to introduce Dr. Catherine Swoboda of Massachusetts General Hospital, and she has been involved in the foundation since 1997. She has conducted various research projects throughout the years and currently is working on retrospective medical record and database review in children and adults with ATP1A3 related neurological phenotypes. In other words, the natural history database. Dr. Swoboda.
Yes, hi, changing gears. Good to see everybody. I have so many questions myself for Dr. George, so I hope we'll have some more time to bring up some of the topics in the later discussions for the meeting. But I thought I'd give an update on the natural history database, what we're trying to do to make it more user-friendly and usable for pharma and the foundation and researchers going forward to get some just really important information that we really need for clinical trial outcomes going forward, and also to just sort of see what patients are out there in our population so that we're appropriately targeting therapies and so on, because there are these specific tests, as you saw, that are going on with specific mutations. But one of the things we keep learning about ATP1A3 neurologic phenotypes is what the huge range of neurologic phenotypes is. And I'm not going to talk about that so much, but there's just been a lot of increasing referrals for cases picked up, that are picked up quite late, many years later, now that exome sequencing is available. And I think that, knowing the mechanisms more broadly of what's exactly happening to gene expression and RNA and all of these things across the range of mutations is going to be really critical going forward for treatment. So that's sort of the context. Helen Chen and Abby Burney and Ren Zhang worked extensively on this project over the past, 15 months or so, sort of trying to bring it back up to speed. And so I want to thank them ahead of time. And of course I will at the end as well. So the project aims for what the foundation awarded us to look at was this retrospective analysis of available medical records and parent-reported data. Pharma really needs this kind of data because there's not quite enough natural history data published out there. And there's a lot of changes that are happening. So we particularly, there's not a lot of longitudinal long-term follow-up. So we particularly wanted to aim at a little bit of an older population to start with, to sort of see, what's happened with these patients over time and try to get a little update for pharma to understand really what's the course of this disorder over time and how it might differ by different mutations and that sort of thing. And the second part of this grant is, of course, to continue to maintain and expand this biorepositories of cells and tissues and other biological materials. And I would like to really emphasize that I'd like to share some of the samples elsewhere. So please feel free to contact me, if there are other investigators out there that are interested in using these samples. I know that each lab is focusing on a specific focused project. As you heard, Dr. George is now making their own cell lines and Dr. S. So that's good. So having these resources in multiple different places is actually critical to the success of the biorepository project going forward. And I think that technologies will continue to improve and change in that regard. So basically, just to remind people, we've been using the Redcap platform now for probably, at least the past 6 years. And the reason we chose Redcap is that it's one of the most prolific database programs in use, and it's many universities, academic medical centers have it at their institutions, but there are also ways to export the data easily, and it's kind of a universal platform that other clinical investigators have adopted. So it's relatively inexpensive because academic institutions have taken it upon themselves. It can be transferred to other sites with data analysis, and Ren Zhang has been instrumental in helping us maintain this data base. We have the usual basic data entry, demographic data, where's the patient from, and what's their racial background, where do they live currently, all that sort of thing, whether male or female, et cetera. Then pregnancy and birth history, which, because so many of these the more severe phenotypes, so with infantile onset, present in early infancy, even within days of birth or the day of birth. I think that will be more and more important as we think about how to rescue patients, because with other inherited genetic disorders, we are thinking now about, what do we need to do even in the prenatal period? Is there something that we can do? And, are there things that we're going to be able to implement right in newborns if we end up going to newborn screening? This is, again, down the line, but important to have that data to understand that history. Family history, developmental history, the age of clinical and genetic diagnosis, all the usual. Medical history and follow-up is very complex in these patients. We've tried to break it down into some various domains. Again, one of the issues is making sure that we get a core bit of information. The exam changes, the history changes over time as to what's a primary problem. It might be, epilepsy at one point, it might be behavior at another point, it might be developmental issues, et cetera. All of those domains are important. What kind of diagnostic studies and imaging studies have they had? We're still finding that right now, MRI is not being used in regular follow-up, and in some of our older patients, it's been hard to get follow-up MRIs performed because the need for them for usefulness on a clinical basis has not been clear. But I will say that looking at from the patients that we've studied, using at least the 3 Tesla MRIs, there are specific changes that we do see in certain brain regions that I think will be critical as a biomarker to tell us what's happening and going forward. And I hope that we'll continue to be able to incorporate those and get pharma eventually to incorporate those in their protocols as we're studying patients, because I think they are important. Adverse events occur very frequently as patients, often because of medication interactions and so on. That is going to be one of the most challenging tasks, but it's very important to continue to focus on that, because again, once a new treatment becomes available, we have to know what were the adverse events these patients were already having, so that we don't blame it on the new treatment and then ruin a good therapy, because this was already happening to the patients. And so that's going to be really important. And then passing current medications continues to be a challenge. Seeing these patients reliably, excuse me, Luke, help, over here. All right. Go in here, you're going to go in here. Sorry about that. Of course, she never barks, except when I'm on virtual meetings. So I apologize. Passing current medications continues to be an issue because many times, especially early on, when patients are having a lot of episodes, there is a lot of turnover of medications and lots of different people prescribing different medications, and the overlap, and it's still really hard to capture the reasons why one medication was stopped, another one started, and what was the data to support that this medication was useful or not useful, and in particular for which symptom. Was this medication used for behavior? Was it used because there was suspected epilepsy? Was it used for spells? Was it used for a cardiac reason or developmental, an attentional problem? Because even though these medications are targeted to specific domains, they often have interaction with other medications the other patients are on, and they also have potential interactions with any new medication coming forward. So capturing all the current medications, but also past exposures is really important in this population. So this is just a a current age breakdown of subjects in the database, in the newly revised database, which is now has 128 subjects. And again, we focused on patients that we had originally enrolled in the original natural history database. So you can see that in comparison to other databases out there, we have a higher proportion of patients that are in this age range of 10 to 40 years of age. And that was very purposeful because we really need to understand, what's happening. That it's, of course, we're collecting prospective data in real time for the younger patients that are being diagnosed now, but those are being collected now in electronic medical records, which are going to be much easier to access and get data from, because that's really become the new way to do that. Whereas some of these older patients, we're still relying on paper records and from multiple institutions. And so trying to capture that longitudinal natural history data is a huge effort, a huge labor of love, I should say. If you look at the ATP1A3 variant frequencies across the patients in the database, this gives you the numbers that we focused on. And again, this was very purposeful because the E815K and the D801N are the most common mutations out there that cause the classical infantile onset HC phenotype. It doesn't mean that all the other mutations are not important, but it was the opportunity to actually characterize those particular phenotypes for which we have animal models, cellular models. We really need to look a little bit more at G947K as well. But we elected to really focus in on these groups first for very specific reasons. So age of symptom onset, not surprising because this has been published previously in this group, and you know that we predominantly have the D801N and E815K. So predominantly symptom onset is within the classic 2 years of age, but you can see we have a range of patients with onset of their initial presenting sign or symptom well beyond that, even in a small. Within the other mutations and within the variants that are represented in this 128 patients, this is the full range. Of course, we have D801N and E815K and G947R, but we have a whole host of other mutations that are represented here. And it just shows you the importance of targeting, like as we're going forward, thinking broadly about what do all these different variants do across the spectrum of gene expression, protein expression, mRNA expression, which are targetable, how are we going to target them, and to have a comprehensive picture and understanding about what's going on in the population at large. This would be just an example of a Redcap database, like what we're looking at when we're in the database. It's just a very basic database, but it's got opportunities for repeated visits for follow-up. So you get your collection of initial information, history of presenting symptoms, and then you have capacity for doing medical history follow-up, an active problem list that might change over time. You have adverse event repeatable forms, neuropsych assessment data. Again, we haven't entered a lot of data in our database. We have data that has been looked at in these patients from Josh's studies from years ago, but in terms of how to enter that data and which parts of the data to collect, that's an ongoing question that we're going to want more impact interaction from the community to know, what are the instruments we're going to use? Because in truth, many different instruments have been used across the spectrum. Many different types of providers have done the assessments. Some are school assessments. Some are much more detailed assessments. Some are specific types of tests. Some target behavior. Some target sleep. Some target, learning and memory. Some target attention. So that in itself is could be a whole entire separate domain that really we have to focus on. I don't think that's going to be the primary target of clinical trial, the initial clinical trials, which is why we haven't put as much effort on that particular piece. And then the current medications and supplements, which that alone is the full-time job for, if we could call patients, every single week and stay on top of this, but it's almost impossible. It's because things are changing, patients are having spells, new things are being tried. So with each visit, which we were able to do virtually, which was a huge blessing over almost the entire of 2020. We were able to do these repeated visits and enter this data. That was really helpful. Now things are changing again, and our institution, I know Duke never let virtual visits really happen. I know that our institution now is completely cut off virtual visits, and that's a huge loss to our community because these kids and adults do not tolerate complicated clinic assessments well. And it's a huge burden on the families and parents. And we just need to talk about that aspect a little bit more as a community. I think advocacy for virtual visits, for clinically important information for helping the management of patients is going to have to be a point of advocacy going forward. That's a really important topic for this particular disorder, which, which isn't quite getting the attention it needs because virtual visits are being continued within states for common disorders, but for rare disorders, it's the out-of-state licensure and the medical risk of medical malpractice and other things that are sort of consuming the community. So that's a little bit off topic, but I wanted to bring up that point while we were there. Here's an example of, a subject where we've kind of put in multiple levels of different information, different adverse events that have happened, 15 in this case, different medications and supplements and changes, et cetera. That's really more the same. So we've gone back and forth about how to collect best over the years, this episodic information. And honestly, the best data still comes from the parents painstakingly noting down on a calendar on paper sometimes, what's happening in a given day. But we had started initially with a longer questionnaire, and we ended up thinking that the burden on the families, people were willing, and we have a number of individuals who were able to do this for weeks on end and continue to do this, but we decided that it was not going to be easy enough to use. So we went to this 4 questions via an automatic survey link, which has been much easier for parents to do, but of course there's less detailed information. And it really has just 4 questions. Did the individual have 1 or more AHC or seizure-like episodes today? Yes or no? If they didn't, it stops. If they did, that would be a yes. And then you would go, was it an HC or a seizure-like episode? And I specifically list it still as seizure-like because we don't always know. But I do think it's still of value to try to distinguish those types of episodes. HC means any other episode, and they're defined in our initial description for the families. Like that could be a hemiplegia, hemidystonia, a full body, eye movement abnormalities, other, right? And then did you use rescue medication? Because we think that's important. How often are the families having to use rescue medication? And then how many total hours was your child unable to function normally today? And we have a range. So I'll show you how that works. And this is, again, best used in patients who have frequent episodes or clusters of episodes where they're willing to say no on the days they don't have it, but they're having enough episodes that they're willing to keep doing it day in and day out for weeks on end. And I think that population is going to be one very early that's going to be targeted with clinical trials because pharma is going to view that as something that's potentially changeable. Whether that's the right approach or not, we don't know. So here's an example from a very challenging young subject with an E815K mutation. This child has all sorts of different episodes, HC, seizure-like episodes. This represents a total of 120 days of data. Out of those 120 days, on 54 of those days, the individual had 1 or more HC or seizure-like episodes. The answer was yes. And then going on to characterize those, how often did you use rescue medication? Yes versus no, in terms of discrete times of rescue. And then how often was it an HC or a seizure-like episode, or both on any given day. And then if they were having spells that day, how many total hours per day was your child unable to function normally? We just left that very vague. Like, how is it impacting them in some way? Less than an hour, between 1 to 3 hours, 3 to 6 hours, more than 6 hours. So I do think it's very simple. It's short. It has limited data, but it's the kind of data where if you started a new medication, like a new seizure medication, for instance, in this child, it becomes very obvious when something's working versus not working. And you can figure out which type of spells does it seem to be impacting, yes or no. And then, you know, are they able to function within their normal range, right? Not specifying that. So I do think this kind of a tool is still critical. We still don't have a consensus on what kind of a tool exactly we're going to use going forward, but I think this is useful. This is an example of how these daily episode logs and medications information are valuable over time. This is very early on in the database years ago when we were collecting, and one of the patients who we've now had, 20 years of follow-up. So just give you an idea of, basic summary of information. This is painstaking to kind of put these things together, but this is also data that parents are normally collecting. They're saying, they're writing down on their calendars. We don't want to lose that data because it gives us very important data over a very longer period of time. So over a period of, from February 2002 to September 2003, we got an idea of what was happening in a given month, what new medications were started or stopped, et cetera. And again, it's very hard to do this in a natural history database very well without the current database setup we have, because these things have to be documented and captured in real time. So save those calendars, save your notes. They're still invaluable because I guarantee you that for most of you out there, all the changes in your medications, you're the ones that are keeping those. You're the ones that have documented them, and nowhere in any medical record is it as good as what you capture. So I just want to reiterate how important the community is in capturing this information, even if it's basic, full body versus, something else. That's very basic, but it tells us what's the main problem this individual is having, and what medications are being added and started and stopped, right? So again, this is looking again at the age breakdown of the subjects, just to kind of remind you that we have a fairly equal representation of male and female in this range. Just want to point that out. In terms of the biorepository progress, we have been we've continued to collect different types of samples because we're not sure what's going to be most useful going forward. And we have collected some recurrent serum and plasma samples on patients on Flunarizine in particular, because we have a collaboration where we're going to share some of these samples based on what dose and the weight of the individual with our colleagues at Xenon Pharmaceuticals, because they are actually at present looking at the impact of Flunarizine on an epilepsy cohort with absence seizures, because that would be a broader indication for Flunarizine and would be easier to get FDA approval. And so we've had serial serum and plasma samples for those patients. We have 94 new DNA samples, 23 RNA samples, which were collected and processed via Paxgene tubes. We have not been routinely doing any CSF studies. And we've continued to establish some skin fibroblast cultures, but as Al George mentioned, there are other technologies now that you can make these, iPS cell lines from blood, and skin fibroblasts are kind of expensive to make and maintain. So we need some feedback at some point, and as what is the consensus for what types of samples we should be focusing on going forward. And we really stopped collecting any more EDV transformed cell line. That was something that we did for 15 years. And we had dozens of EBV transformed cell lines, but since that process of transforming the cell line introduces Epstein-Barr virus-induced, mutations, it turns out they're not as useful to use for projects going forward. So I'm just going to briefly, in the last little bit of time, because I'm really out of time, just highlight the capstone study, this questionnaire that many of you did for Abby Burney. I'm going to go through this really, really quickly. And just because I think it was an important participation by the community, I think it leads to some important knowledge about Flunarizine to try to get that approved. So the whole purpose of this study was to assess parent perceptions of Flunarizine to treat episodic neurologic symptoms in AHC. And what was spurred this project was basically the fact that Flunarizine is still not approved in the US, even though we know it works in some patients, but we don't know which patient works on, and we don't really have the right dose yet worked out, and we don't have a way to monitor it properly. So there's lots of reasons. So I'm gonna, this will be published hopefully in the not too distant future. It's currently being reviewed in neurology genetics. So I'm just gonna highlight a couple things. You know, just to remind people that Flunarizine is used worldwide to treat a lot of different things, including vertigo, cerebrovascular blood flow insufficiency, prophylaxis of migraines. It's a really complicated drug. It has lots of functions. Still don't know all the things it does. Off-label prophylactic treatment since the 1980s in the US with efficacy reported by parents ranging from 30 to 78%, depending on what little review you look at. And the E815K common mutation mouse model by McCarty was specifically studied, and he felt that the episodes of seizure went down with the E815K model. But the long-term effects aren't well understood. We don't really know what the adverse event profile. It's not FDA approved in the US for any indication. It continues to be a problem for us to get it. So these were some of the research questions. And again, I don't really have a lot of time to go into this, but it's just keep in mind that we were trying to get at specific questions. Does perceived efficacy change over time? What treatment factors are associated with increased perceived efficacy? Does genotype impact efficacy? Were there adverse events when stopping Flunarizine? And what are parents' perceptions? Are they interested in future controlled trials? Or are they just done? Have they decided? And then are there barriers? So Sharon and Lynn, I'm going to interrupt here and say, I don't want to talk about all these results because I think we can better use our time. But if people want to know more about this going forward, but in a nutshell, whoops, it's not letting me go forward now.
She seems to be frozen.
Yeah, I don't know what.
Then you got about 5 minutes or so before lunch and questions. So.
Okay. All right. So let me see what's happening. I don't know why it stopped. All right. So I'll just keep talking about it and give my acknowledgements this way. So basically, the bottom line of this study is we found some very interesting things. Number one, you know, the majority of patients who have stayed on it for many years, I mean, stayed on it because they perceive efficacy. And we have some data that when they went off it, that episodes worsened. But the data is all about perceptions, right? And it's very hard to do this in a retrospective way. But E815K, notably, patients, parents of patients with an E815K reported the greatest perceived benefit in reducing frequency, duration, and the greatest reported efficacy. And I think that fits with what we know from the animal model. And talking to you know, investigators who've used Flunarizine for many years, it is an adjunct agent for epilepsy. It does treat absence epilepsy and so on. And I think it might be providing adjunct support in that regard, based on some of the data we have in the database. So I think that we gained some very important, valuable information. The purpose of publishing this data is that it doesn't work for everybody. We did see reported adverse events that led to stopping of medication in a number of patients, but they were very poorly documented as to why. And it's not clear how much that was dose related or other things happening at the time. And in almost no case is there really good documentation in the 3 months before and after Flunarizine for like exactly how it changed episodes that we could easily access at this point. But the point is that the community feels it is helpful in some cases. We have some data in the animal model that's useful. We have previous studies that have supported its use. And now this paper is the sole purpose of this paper is to try to get it FDA approved so that we will have access to it to use for trying it in our patients in an appropriate way, and ideally via another study, and particularly in the youngest patients for which the dosing, the start of the dosing is not clear at all. And we know that there's a lot of variable metabolism of Flunarizine based on different variants and handling of the drug. And we know that it interacts with other medications. And so the purpose of this paper is to get interest and the importance of how it's negatively impacting our population to not be able to get it through an appropriate venue in this country. It leads to misuse by physicians who aren't comfortable using it or who don't have an IND. It leads to delay in access of the medication because people are reluctant to start with it. It leads to uncertainty in newly diagnosed parents of newly diagnosed patients because they're not sure why it's not approved by the FDA. So for all those reasons and more.
We lost Dr. Swoboda altogether. Well, thank you, Dr. Swoboda, for your talk.